So today we're going to talk about lecture 3. Again, none of these are really like hardcore chemistry or hardcore biology yet. A lot of the stuff that we're talking about are still more or less reviews of general chemistry, things that should not be too non-familiar to you guys. Right? So you guys should have seen these things in one class or another. Okay, so it shouldn't be that hard. All right, so the stuff that we'll talk about today, minus the iso lecture point that we'll talk about on lecture 4. So we're gonna very briefly talk about amphipathic compounds. Okay, so what are those, and we'll go over those in a little bit. We'll talk about primarily ionic equilibria. Okay, so that's sort of the topic for today. And then we'll get to Henderson-Hasselbalch equation. Okay, so that's the equation that we'll definitely use again and again to look at the relationships between pKa and pH of the medium. Okay? Very quickly, amphipathic materials, those are molecules that have, generally speaking, a polar characteristic and, in addition, a nonpolar characteristics, such as fatty acids. Okay? So amphipathic, it's an important class of molecules that exhibit both hydrophilic and hydrophobic properties simultaneously. Okay? And it is not surprising that you run into these things almost every day during your life. Okay? For example, these materials or substances, they include fatty acids. Okay? You know that your membranes are made out of fatty acids. Lipids, not really. Okay? But here we primarily mean membranes. Okay? So membranes are like that. Lipids in the form of triacylglycerides, not really, okay? Detergents, definitely. Detergents are absolutely materials that would have both polar and nonpolar characteristics, okay? This is actually the reason why you use detergents to wash off grease, like fat or like anything oil on your clothes. You use detergent because let's say you have oil, right? And we know that amphipathic molecules will have a nonpolar characteristic. So you got nonpolar, and then on the top you got something polar, right? So then you can imagine you would have a layer of organics that sort of surround the oil, and then on the outside you got all these hydrophilic functional groups that will attract water. Okay, so detergent, that's how it works. Okay, so of course we have different types of detergent, and some works better than others. Right? For our purpose, primarily we'll see how amphipathic molecules are generally characterized by a polar head group followed by a hydrophobic tail. Okay? Well, that's just how it is, because these things typically have like charges on them. Okay? And then the nonpolar tail, those things have a lot of carbons. Okay? So a lot of CH2, blah, 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 blah. Okay? Like that. Right? Some very good examples that we run into in nature, for example here, you have dodecanoate. Okay? So this is C12 acid. Okay? So you got 12 carbons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay? And on carbon 1, you have the organic acid group, okay, or carboxylic acid group. Right? So because this is an acid, it's going to be able to mingle with water. Okay? That makes sense. Now you got 12 carbons long in terms of a carbon chain, and these guys are absolutely nonpolar. So they are hydrophobic tail groups. Okay? So usually they're really long. Right? So they're fatty acid. That's actually where we got the name from. Fatty, things like they look fat. All right? The second molecule, this guy is called dodecyl sulfate. You will be working with this if you are studying proteins, okay? So I think many of you will have heard of this thing called SDS page, okay? SDS, that's the DS part, okay? DS. And in SDS, this actually is standing for sodium, okay? Because it's a sodium salt usually, okay? Regardless, so dodecyl sulfate, just like the neighbor, your dodeconoid ion, it's also 12 carbons, okay? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 carbons, okay? And on the first carbon, you have a sulfate attached to it. And sulfates, guess what? It's a negative charge, okay? So it's very hydrophilic. So then again, it can interact with water, okay? So you have a hydrophilic head, okay? Always you have a head, and then you have a tail, okay? So those are your amphipathic material, okay, or compound. Sometimes when we talk about membranes towards the end of our class, okay? Towards the end of the class, we'll start talking about membrane structures and that kind of stuff. And what you'll see is for most membranes, you have a head group like that, okay? And then you have two tails like this, okay? And the head group, you have negative charge, positive charge, things that will make it very hydrophilic. So those will attract a lot of water, okay? So obviously membranes, this is going to be towards the outside, and then you'll have the lipids binding with another layer of lipid. We'll see a picture of that just a bit, okay? So there's no problem with the definition of amphipathic material or compound, okay? We're good. So amphipathic molecules, those are very important because in most of biology, we are sort of primarily composed of water, right? So water being the most important solvent, especially it's solvent for life. So if you throw your amphipathic molecules, like a fatty acid, into water, right, what happens, right? I think you guys already know what happens. The nonpolar part of the molecule will bind with each other like this or like that. The polar part, okay, or the hydrophilic part, they bind with each other like that, okay? So if you throw these amphipathic compounds into water, you'll have several choices of the type of structures they form. You can form a monolayer on the surface like this, okay? That's a monolayer on the surface. Or the organics sort of is exposed to air, that's okay. And then the hydrophilic interacts with water. That happens, okay? But more often, what you actually get are these things called micelle and lipid bilayers, okay? Micelles are these things, okay? So that's a micelle. So what they are is essentially you have the hydrophilic on the outside and then hydrophobic on the inside, okay? So that's very straightforward. So you can understand how that would happen, okay? Lipid bilayer is different, okay? Bilayer is essentially you have hydrophilic outside, hydrophobic inside, all right? And then you have another layer of hydrophilic in the inside, okay? And that's just the way it works. You have so you have lipid phase and attract to it another lipid. So you will leave with water on the outside, water on the inside, okay? So there are two different types of structures that amphipathic molecules can adapt, okay? So anyone wants to ask, when do you form micelles and when do you form bilayers? You probably want to ask that, okay? Maybe you don't, but I'm going to tell you anyways, okay? So actually, it turns out, it would have to do with the shape of the molecule, right? So for micelles, typically you have a big head and one tail. So if you look at the geometry, it's going to look more or less like a cone, right? So if it's a cone, then the most probable structures that they form together will be a micelle, because the tail, you only got one of them, and it's narrow, so you will form something like that. So that's like micelle, right? So it's like a cone thing, okay? Like that. That makes sense, right? So bilayer, on the other hand, typically forms when you have a head group and then two tails, okay? Such that the shape looks more like a cylinder. So you will adapt a more cylindrical shape. So it's a more regular type of shape. And because of that, you can sort of form a plane, like a little surface almost, okay? And of course, eventually they're going to close somehow, right? Because otherwise, if it doesn't form a little circular structure, then you would have hydrophobic exposed here, right? So eventually, this end will connect to this end. Okay, that's just because of hydrophobic effect. Okay, remember hydrophobic effect? We talked about that on Tuesday. I think I did. Okay, so these are the two structures or the two types of things that these molecules form, right? So obviously, lipid bilayers are important for cell membranes, right? We talked about the 7 pillars of life and how seclusion, okay, or compartmentalization is it's actually a very important trait for life. So that is used to make membranes, okay? Of course, these things are actually very useful in terms of drug delivery, okay? For example, if you have a hydrophobic drug that you want to deliver to some place in your body, and you want it to be transportable by something that's probably hydrophilic, right? Because your body is made out of water, okay? So what you can do is you can use a micelle, okay? And then you can put your drug or whatever into your micelle. And then outside is a layer of hydrophilic characteristics that allows it to go to different places. And of course, you're going to put some stuff on here that tells it where to go, okay? So there are a lot of people working on these type of drug delivery vehicles to deliver drugs to specific regions in your body, okay? So micelles are also very useful in that way. Also, most detergents that you buy that you use to clean your dinner table, usually they also form micelles, right? Because on your table, let's say you have your table, and then you got like grease all over your table, and then you take detergent. That's my ugly version of a spray bottle, okay? And you spray detergent on top, you wipe it off, then you have the oil trapped in a micelle. And then you apply water to it, and then you can wash it off. Okay, so that's that. Okay, we're good? Everybody can understand this, right? Okay, that's actually quite simple. All right, so that's sort of amphipathic stuff that I want to talk about, right? So we come back and visit the common interactions that we see in biochemistry. So again, you've seen these hydrogen bond. This guy will be a acceptor. This guy will be a donor, because this guy donates a hydrogen to this guy, okay? So same thing, acceptor, donor. Okay, so the donor of the hydrogen bond pair is the one that's donating the proton, okay, but whatever. So you got hydrogen bonds that are really, really important in biology. You have ionic interactions, so they actually form, so these things are called salt bridges, okay? You got a minus charge, you got a positive charge. When they connect, you got a salt bridge, okay? It's pretty stable. It's a stabilizing effect, okay? So that would be an attraction. Of course, sometimes you have repulsion, okay? That happens when, let's say, you have two positive charges really close to each other, they repel. So that also contributes to protein structures sometimes. Okay? All right? So this is charge-charge interaction, and these are essentially dipole interaction, but it doesn't really matter. Okay? All right. And then we also got hydrophobic interaction. So we know that the hydrophobic stuff tend to come together, okay? Because you want to minimize the surface area between hydrophobic and water. Okay? And that is because of the hydrophobic effect. And last time we explained how hydrophobic effect is an entropic effect. We did, right? Okay? So that's fine. Right? So you guys should be, you definitely need to be able to explain hydrophobic effect. Okay? So that just comes up everywhere, right? So you have to be able to do that. And then you also get some like van der Waals interaction that happens when two atoms get close to each other, but less important. Okay? Primarily, you really need to know hydrogen bond, ionic interaction, hydrophobic effect. Okay, hydrophobic interactions. Okay? So those three are the most important interactions that we see in biochemistry. Okay? There's one extra, this slide, I didn't know where to put it, so I'm just going to put it here. So it turns out a lot of times when we say, how you have a proton in the water, right? Turns out the proton itself does not really move like a lot. What happens is here you have H3O plus, right? The proton on the water, it can hop between waters. Okay, so the proton itself, so this H3O does not have to physically move. Okay, all you got to do is this proton can go from H3O to this H2O. You turn this guy into H3O, and then this H2O passes on. You turn it to H3O, so on and so forth. So the proton can actually hop between water molecules. So actually, with this ability, you can very quickly pass your protons from location 1 to location 2 very quick. Okay, so this is almost like a super quick effect. And towards the end of the class, when we talk about sort of like protein or enzyme mechanisms, we'll get to how sometimes in an enzyme, okay, the effect felt by this water can translate up all the way to this water, okay, through the whole proton hopping effect. Okay, so that's just a little thing that I want you guys to know, that waters, okay, or at least protons, okay, hydronium ion does not have to move. Okay, all it has to do is pass the proton from the hydronium to other water molecules. Okay, and then essentially the proton will move. Okay. All right. So here's the little conclusion for everything that I talked about on Tuesday and up to now. Okay, is that non-covalent interactions are very important for the structure of macromolecules. Okay, that's very important because they determine the 3D structure, whether or not they are held together or they repel each other. Also, these things are very important for many of their functions, okay, such as enzyme catalysis, as we saw on Tuesday, and for DNA recognition, as we saw on Tuesday. Okay. Number 2, charged molecules, they form salt bridges and charged dipole interactions that are stable provided that you have plus and minus, or let's say OH and minus, okay, or positive and OH, something like that. Okay? So those things are also going to form you interactions that will stabilize the overall energy. Okay, so these things want to form. Okay. Number 3, water has permanent dipole moment. That means that it's a polar compound. It participates in significant amounts of hydrogen bonding. So you should be able to explain some of the features of water, like the density, like the boiling point, that kind of stuff, okay? So you should be able to explain in terms of hydrogen bonds to those characteristics, right? Lastly, and most importantly, hydrophobic effect is entropic effect, okay? So you should really know what this means, okay? What this means, okay? So essentially, again, is when you have individual droplets, oil, so this is oil droplet, okay? You will form cage-like structure, okay? So cage-like water, okay? So that the entropy of water is low, okay? And remember, if then you turn all the the oil droplet together and you form a ginormous oil, then you minimize the surface area between water and the oil droplet. So your entropy of water is less low, so the delta S actually increases. Okay, so this is the hydrophobic effect. All right. So today, really, I want to talk about ionic equilibria. To some of you guys, this might be like boring. Okay. So in biochemistry, really, we have a simplified version of acids and bases. Okay, so for most of our purpose, we will be satisfied with the Bronsted-Lowry definition of acids and bases. Okay, so essentially what it means is acids are proton donors, bases are proton acceptors. Okay, so that's like very simple for biochemistry. Of course, in organic chemistry, you usually take the Lewis acid and Lewis base definition of acids and bases. Okay, but for biology, most of the biochemistry that we will see, at least in the undergrad level, we usually just pretty happy with the Bronsted-Lowry definition. Okay, so again, a strong acid is something that dissociates almost completely into hydronium and a weak conjugate base. Okay, weak conjugate base is something very stable. Okay, for example, Cl minus. Okay, weak acid partially dissociates, meaning that you have some kind of equilibrium that forms between the dissociated and the non-dissociated forms. Okay, and then strong base is the vice versa for releasing OH minus, such as NaOH gives you Na plus and OH minus. Okay, and OH minus is sort of a powerful proton acceptor, so you can grab protons to it to generate water. Okay, and then a weak base obviously grab a proton with an equilibrium with the protonated and the non-protonated form. Okay, so that's his definition of strong acids and bases. Okay, so water. Okay, so like I said, when I was undergrad, I spent like 2 months on water. That's kind of crazy. So we're only going to spend like 2 minutes, 20 minutes, something like that. All right, so water, all right, can form hydronium and hydroxide ions. Okay, that's actually an equilibrium. Right, so you do this all the time. You go from water, you split that into OH minus and H3O plus. Okay, so it's an equilibrium. And of course, we like to do a little math here and there. So we say, oh, okay, you have an equilibrium constant for this reversible ionization of water is equal to the equilibrium constant, KEQ. Of course, product on the top, substrate on the bottom, okay, or reactant. And you got H plus times the concentration of H minus divided by 55.5 molar. Yeah, this guy is the concentration of water in water. Okay, how do you know that? That's because the mass of 1 liter of water is 1,000 grams. So you know the molecular mass of water is 18 grams per mole. So then you take 1,000 divided by 18 grams per mole. You get 55.5 moles per liter. Oh, wow. Okay, that's cool. So this is the concentration of water. So then you rearrange the equation a little bit. You get this equation where you have the equilibrium constant times 55.5. We then just call that Kw, the constant for water. Of course, then we can use some electrical conductivity experiment, and we will know that the KEQ for pure water at 25 degrees C is going to be about 2 times 10 to the minus 16. Okay? So you can plug that number in, all right? Plug that number into kW, and what you get is about 10 to the minus 14 meters squared. All right? And then to make it easier, essentially, okay? So we can say that you can use this value, okay, and then you can calculate depending on if you have the H plus or the OH minus, right? So regardless, to make it understandable, we usually define pH, okay, as the negative log of proton concentration. Wow, okay? So that tells you that a pH difference of 1, okay, gives you a difference of 1 order of magnitude. Just always remember that. Okay, so pH 1 and pH 2, they change in terms of acid concentration by tenfold. Okay, okay, so we're cool. All right, so this is just water. All right. So pH. Let's take a quick look at some of the common stuff that you find in your house and what are the pHs for those solutions. At the very top, you got NaOH and HCl, okay, because 1 molar of HCl will give you pH of 0, okay, because you got 1 molar of proton. Somewhere in between, you got lemon juice, you have Coca-Cola, vinegar, that kind of stuff. You have tomato juice, red wine, beer, black coffee, okay, so those are pretty much acidic stuff. And then when you get towards neutral, you have your saliva, okay? You got milk, that's around neutral. Human blood, we'll see today, it's about 7.3. And then gradually you go into more alkaline or more basic. You have seawater, egg white, you got baking powder, things like that, okay? So those are the spectrum of the things that you see. Now, what you notice, a lot of things that we see in biology, they tend to be more acidic. Okay, and usually that is because organic stuff will be like they have carboxylic acids. Okay, anything that have carboxylic acids are in general organic acids, so they lower the pH of your solution. Okay. All right, so I'm not sure if you guys need to practice this, but let's go over this real quick. So what is the pH of a 0.1 molar sodium hydroxide solution? Okay. So again, we know that sodium hydroxide dissociates completely because it's a strong base. We know that the Kw is equal to concentration of proton times concentration of OH minus. Okay. You rearrange the equation and we know Kw is 10 to the minus 14 molar. You know the concentration of OH minus because I told you right here. So you divide that number, you get a hydrogen, or not hydrogen, your proton concentration 10 to the minus 13. Take the negative log of that value, you get pH of 13. Okay, so that's this very simple thing that you guys should know, right? So ionic equilibria, right? So when we're talking about ionic equilibria, okay, we are concerned primarily with weak acids, okay, because most of the biological acids are weak acids, okay, so they will have an equilibrium, right? So for any acid, general HA, right, they can dissociate into proton plus the conjugate base A minus, okay? You can arrange that into their equilibrium constant, KEQ, being concentration of proton, concentration of conjugate base, divided by the concentration of the acid itself. Okay. Of course, we like to give it a different name. We just call it the acidic constant, so Ka. Okay, Ka, because we know that it's for an acid, right? And pKa we can define using the same logic as we would for the concentration of proton. So we can say pKa is equal to the negative log 10 of Ka. Okay, so essentially it says the lower the pKa, the more likely that proton is removed from this compound, because the lower pKa, then that means higher the Ka. If Ka is high, that means that the proton concentration is going to be larger than the acid. That tells you that the acid wants to make protons. Okay? Well, cool. So this is a table of some of the common organic, inorganic acids that you see around biology. For example, you have formic acid that can go into formate anion that give you a pK of around 3.75. If you add one more carbon to it, okay, you make it into acetic acid. That's the vinegar that you drink. The pK actually increases by 1. Okay, that's actually very interesting. Right? So we'll talk about that later. Or lactate. You generate lactate when you go out and exercise in your muscle. Lactate is also very acidic. It has a pK of around 3.86. Okay, so what you can see is that most organic acids, so all of these are carboxylic acids, right? They have pK of around 4. Okay, so around usually 2 to 4, but usually it's around 4 if it's in the open aqueous solution. Okay, so around 4. So that means that when pH is 7, okay, if the pH is 7, right, your acid is usually in the conjugate base form, okay, because your acid wants to throw the proton to water, right? And then you got some other things like phosphoric acid and phosphoric acid minus 1 proton, phosphoric acid minus 2 proton, phosphoric acid minus 3 protons, okay? And obviously you see that the first proton to pull off is very easy, okay? pK is 2. The second proton becomes harder to pull, the pK of 6.9 roughly, okay? And then the third proton is even harder with a pK of 12.4, okay? Why is that? Okay, that is because every proton that you remove, you are accumulating charge on this molecule, so the molecule becomes less happy, okay? And then as a result, the pK becomes higher, all right? Okay, so really the stuff that will really concern us is the Henderson-Hasselbalch equation, all right? Where the Henderson-Hasselbalch equation describes the change of pH during the titration of a weak acid or a weak base. Okay? So this is the H&H or Henderson-Hasselbalch equation. pH is equal to the pKa of an acid plus the log of the acid's conjugate base divided by the acid in the acid form. Okay? So we're going to derive this equation. The derivation works like this. We know that Ka, okay, the acid constant, is equal to proton concentration times it by the conjugate base divided by the acid. Okay. We solve for proton concentration. You rearrange, and you get that. That's cool. Okay. And then you do the negative log of both sides. So you do negative log on here, negative log on here. So you get this. Negative log of proton concentration happens to be pH. That's cool. And then when you do negative log of this entire thing, you can separate it out into negative log of Ka plus the negative log of the acid over conjugate base. Okay? So you can rearrange that. Right? And negative log of Ka happens to be pKa. Okay, and then minus the log of acid divided by conjugate base. Okay, now we don't like acid to be on top, okay, because we're always used to having product on top and then substrate or reactant on the bottom. Okay, so we flip it and then you get pH is equal to pKa plus the log of proton acceptor over the proton donor. Okay, so this is going to be A minus over HA. Okay, so this is the Henderson-Hasselbalch equation derivation. Okay. You don't have to remember how to derive this thing, even though I don't think it's that difficult, but you have to know how to use it. Okay? Now, let's take a look. So let's say I start with formic acid. This guy is formic acid. Okay? I start with formic acid. right? Let's say I do 1 molar formic acid. The initial pH is going to be pretty low. It's like around 2, something like that, all right? And then I start to add base. I start to add things like NaOH to the base, to the solution of formic acid. And immediately you see a rise in pH, okay? However, gradually the change in pH slows down until where you hit a special concentration of 0.5. You add half amount of NaOH to your formic acid, then essentially what you get is half of this and half of this, okay? So you go back and you look at your Henderson-Hasselbalch equation. If the top and the bottom are equal, essentially you cancel this out. The log disappears. Log of 1 is 0. The pH then is going to equal the pKa, okay? So this is how you use to titrate, or at least you can find the pKa of a unknown organic acid, okay? So let's say you have an unknown organic acid that your professor wants to identify what it is. You can first identify the pKa of this organic acid by using a titration curve, okay? So you have a pure acid. Of course, you have to assume it's pure, okay? Which is kind of weird, like who would assume it's pure? So you would have to first have some way of separating the different chemical species. Let's say you separated this unknown organic acid. You want to know the pKa of this organic acid. Then all you have to do is start titrating it, okay? And then every time you titrate it, you monitor the pH of the corresponding solution. And then where you see a flat, so at this moment, the slope is actually 0, okay? Then at this particular point, that is your pKa for that acid, okay? Same thing happens if you have ammonium, okay? So ammonium is a protonated form of ammonia, okay? So you can actually get ammonium chloride from companies. So if you dissolve ammonium in water, initially the pH is going to be relatively low, okay? And then you add, same thing, you add sodium hydroxide, and then gradually, first you have a sharp increase in pH, and then gradually your slope decreases. Eventually you reach a slope is equal to 0. This is the pKa of NH4 plus, okay? So it tells you the pKa of NH4. Have you guys generated this before? This is actually pretty cool. Like when I was an undergraduate, in my general chem lab, so like we got 20 students, right? And 20 students, so the professor gave us individually like 1 amino acid, okay? And then we're responsible of finding out what amino acid we got. And the first thing that you can do is measure the pKa of your amino acid, okay? And for very quickly, how many pKa's will amino acids have? 2, minimum 2, okay? Some amino acids will have 3, okay? Well, cool. You guys know where we got that? Amino acids, right? Amino acids looks like this, right? So you have one group that's ionizable. You have another group, right? So you can have a pKa for this hydrogen. You can have a pKa for this hydrogen, right? So all amino acids will have at least 2 pKa's, okay? So we'll come back to the next class, okay, when we talk about polyprotic acids, right? So this is Henderson-Hasselbalch equation. All right. Henderson-Hasselbalch equation is super useful when it comes down to buffers. Right? So hear me out. Okay, as a professor in university, okay, as a PI of a lab, there is one thing, I have 2 things that we really, really wish our students know before they join a lab. Do you have any idea what that is? Well, number 1 is the ability to make solutions. Okay, turns out, okay, don't laugh, but turns out it's harder than you think, okay, to most people. I don't know why, okay? You need to know how to make solutions. That's one thing. Another thing is you need to know how to make proper buffers, okay? Because in biological research, buffers are essentially everything, okay? So for every type of chemical reaction or any type of biological reactions, you're going to use buffers somehow, okay? One kind of buffer or another, okay? So what are buffers, okay? Buffer solutions are those solutions that are able to minimize the change in pH following the addition of acids or base, because the conjugate acid, HA, and the conjugate base, A-, of the buffering compounds are present in sufficient concentration to combine with the added H+ or OH- and neutralize them, okay? Yeah, that's just a lot of words and you guys don't understand what that means, okay? Essentially, what you want to know is this, okay? All right, check this out. We know around pKa, okay, pH fluctuation is low, okay? And how do I get it to around pKa? The pH of a solution if I use a chemical buffer. That is when A- equals HA, right? So when A- equals HA, this term drops out, pH equals pKa. Okay, we're cool. So let's say when your A- or your HA, it's in enough quantities, okay? Small fluctuation, like you add a little bit of HCl, it will not change too much of your pH. Why? Because let's say you have A- equals to HA, right, initially, and then you add a little bit of HCl. What do you make more, A- or HA? HA, right? You add a little bit more HCl, you make more HA. Makes sense. Now, let's say you increase a little bit of HA, okay? But since we're taking a log of this number, okay, a little increase, and then you log it. Is that increase going to be large or small? It's going to be pretty small, okay? That's why when you see it here, when your pH is at the pKa, okay, when let's say your HA equals to A-, okay, and you add a little bit of acid or a little bit of base, you change the concentration a little bit, but because you're logging it, okay, the actual change on the pH is minimized, okay? It's minimized. So this is what makes it a buffer, okay? That's what makes it a buffer, right? So buffers are really, really important because a lot of times we're doing enzyme reactions, okay? And enzymatic reactions, a lot of times, will release charges, okay? Will release, for example, protons or phosphates or whatever. So things that will change the pH of your medium, okay? So buffers are really, really important in biology, right? And knowing this, okay, knowing the Henderson-Hasselbalch equation, okay, so this will help you to decide which buffer to use for your chemical reaction or your enzymatic reaction, okay? Generally, you want to pick a buffer, okay, the chemical, that has a pKa of the pH that you want to do your reaction at. Does that make sense? Okay. Let's say I want to do my reaction at pH 7, okay? Then it is in my best interest to find a chemical that has a pKa of 7. Okay, that would be the best. Okay, that would be the best. However, we know sometimes that doesn't happen because, you know, how do you like just magically find a chemical that has pKa of 7? Even if you do, okay, even if you do, guess what? Sometimes your buffer can interfere with the enzyme activity, okay? So unfortunately, sometimes the buffer may actually inhibit your enzyme, okay? So as a result, biochemists will develop tons of different buffers, okay? And they have different buffering range, okay? Now, this is a very useful table that I got, I think, from the internet. There's the citation all the way at bottom, so I'm not copying people, but maybe I should tell them that I'm borrowing their chart. Yeah, but you can find a lot of these tables online. For example, Sigma will have their own chart. Most chemical vendors will have a chart that serves as a guideline for you to think about what kind of buffers that you want to use when you're doing your enzymatic reaction. Okay, so what you can see is buffers. Typically, we don't really look beyond lower than like 4, 5. We don't care because enzymes don't really work in that range, generally speaking. Okay, same thing with above 10 and 11. Not so many buffers that we care about because enzymes don't usually work in that pH. Okay. So we usually care about 6 to 8 to maybe 9. Okay, so you got a lot of different buffers. Okay, some buffers are inorganic, some buffers are organic, some buffers you can use with cells, some buffers you can't. Okay, so this is just a chart that shows you you have a lot of options. But of course, if you want to know like exactly which buffer to use, you have to look it up. You look it up, look at the characteristics. Okay. But what I can tell you right now is, for example, tris. You guys know what tris is? You use tris like a lot, okay, in molecular bio labs. Right? For example, every time you want to run DNA, you prepare this TAE buffer or TBE buffer. T is tris, okay, it's tris base. Right? Now, generally speaking, if you don't know like the exact buffering capacity, for example, you see how some buffers have small range, some have larger range. In general, if you are not so sure about the buffering range of your particular chemical buffer, okay, as a very rough, okay, I say very rough, don't quote me on this, okay, very rough general guideline is the pH range, the buffering range is about plus or minus 1 of the pKa, okay. You can sort of use that as a general guide, but if you want to be more careful, it's going to be plus or minus 0.5, plus or minus 0.3. Okay, so like I said, again, the best thing you can do is to find a chemical that has a pKa that has the pKa of the pH that you want to do your chemical reaction at. That would be the best thing. But if you can't find that, try to find one that is close to the pH that you want. Okay. And then a lot of times, you can sort of guesstimate the range of a chemical buffer roughly being pKa plus or minus 1, okay, roughly. Okay?